make the colon.
It's up to you.
It's recorded, so we may as well get into it. So, central dogma. Now, when did you guys start this year ones? Anyone jump in?
Pop it in on the chat.
Jump in chat.
In chat. You're ready to go.
Last week in Oxus.
Cool. So you would have had like a lecture from the French guy, and then maybe an online active learning or something, or maybe that's coming up this week. I know for us it felt very rushed, and for anyone who didn't do bio or maybe didn't complete the whole of the bridging course, it was a bit of an ask, like to cram so much knowledge into just a week. But hopefully I've condensed it enough and it's a bit more straightforward than may have been taught to you guys. Oh no, here we go. So central dogma. Now this little flowchart here is the entirety of like the topic. So even if you don't remember anything else by the exam, if you have this little flowchart in your mind, that should be able to guide you towards the answers of some of these multi-choice questions. So we start with DNA replication. We go from DNA to RNA. Then we go from RNA to our polypeptide, and that polypeptide is then changed until it becomes a protein. So DNA replication. Now, you may have seen this in the active learning or something similar to this. I'm pretty sure I've taken this diagram straight from the Moodle book that we had, we learned from. So it's pretty complicated. We've got these two little blobs here. Those are our DNA polymerase, pardon me. And you can see they're building from 5' to 3', or they're reading from 3' to 5'. Now, as I just did, I got confused, but stick to one of those, either what the enzyme is reading or what the enzyme is creating. So for me, it's easier if I say we're creating from 5 to 3, but for you, it might be, it's easier to say that the enzyme reads from 3 to 5. Stick with one of those and it'll make your life a lot easier, so you're not thinking of two different things and getting them mixed up like I did for about 4 weeks. Anyway, it looks like this DNA polymerase molecule is moving in opposite directions, but in reality, if we've got a double strand, we've got our DNA polymerase like going along that blob like that. So we get this kind of separation between one which is going along a leading strand, going straight from 3' to 5', and then we get this other molecule which is doing a little bit of 3' to 5'. Then it comes back and does a little bit more 3' to 5'. So if we have a look now, cool. So this is our kind of, I don't know, scaffold for our DNA replication. So there's a couple of enzymes in here, and I've got those down the bottom here. So of these answers, what unwinds DNA?
Pop your answers in the chat.
Helicase. I'm liking the the differences in capitalization there. Cool. So helicase unwinds DNA. Spoilers. DNA polymerase then attaches to what? To the template strand. RNA primers. Awesome. So RNA primers are just little blobs of RNA which are complementary to our DNA, and they'll stick there with their H bonds, their hydrogen bonds, and then allow the DNA polymerase to kind of move along there. So 3, DNA polymerase reads the template strands in which direction? So we were just talking about this reading versus creating. 3' to 5'. Thanks, Enoch. Good stuff. And I hope everyone playing at home is also getting these as well. 3' to 5'. Due to this, we get two different strands, X and Y. So one which is just continuous, and then one which is kind of trailing off a bit. So what are those two strands called? Leading and lagging. Good stuff. Ligase attaches some fragments to the lagging strand. And what's the name of these fragments? Okazaki. That was one of like the satisfying names just sunk into my brain. It's fun to say, Okazaki. Then 5, something adds a cap at the end of the lagging strand to complete replication. So what's this cap we talk about? Well, first of all, what enzyme makes this cap? It's the last word we got there, telomerase. Cool. Ah, no, why am I going back? Cool, telomerase adds a cap. And if you've had any HEP lectures, you would have heard Craig Hassard talk about telomeres and how being mindful and exercising is really good for the telomeres and it reverses aging and all of these things. Pretty much telomeres are just, they're the end point of the DNA. So when we replicate it, it doesn't shorten, because if we go back to this diagram, DNA polymerase on the leading strand will be able to pretty much complete it to the end, but the lagging strand, if we don't have a cap there, it won't have a place for the RNA primer to bind and then create from 3' to 5'. So we need these telomeres to ensure that both our leading and lagging strand can be completed in replication. Now, there would have been, I think, an active learning where you would have had to answer a question on telomeres. If you've got any questions, pop those down in the chat or unmute yourself and ask, because that would be a good time to do so.
Maybe raise your hand first if there, so we know who wants to ask questions.
Too easy. Rightio. You're always welcome to chuck other answers in the chat as well. So here we've got, there's just that same diagram again. So 3' to 5', we're reading, creating in one direction. Then 3' to 5', we're creating our DNA, but it's going backwards. 3' to 5', 3' to 5', 3' to 5'. And those are our Okazaki fragments. Cool. Next we get to transcription, and this meme I think I made 2 months ago, so I'm sorry for the poor content, but pretty much it's the same thing, except instead of making DNA, it's making RNA. And if you think about it like that, it may get confusing. For me personally, I find it easier if I've just got the one process and then I'm switching out certain variables depending on what we're talking about. So helicase unwinds DNA. Same vibe. We need to unwrap that double helix so we can get our enzymes in there and do what we need to do. Instead of DNA polymerase, it's RNA polymerase. Pretty good concept there. And it attaches to the promoter region of the gene. Thanks, Ben. So RNA polymerase attaches to the promoter. What is the promoter? Is it at what end of the gene is it? Someone can type it in the chat, please, or raise your hand and put the mic on. So I'll ask again, maybe. The promoter region is, well, RNA attaches to the promoter region of the gene, but where is the promoter region? What end of the gene, of the RNA molecule? So
Maybe give them a little clue.
okay, so we start translation here, transcription here. Upstream, yeah, so upstream of where we start the actual gene. Good stuff. Thank you, Marianne. So we've got our gene of interest here. Now, am I mirrored? This is going to be confusing. Okay, gene of interest here, and then don't need that. This is our little promoter region here. So before the actual gene of interest there, RNA polymerase will bind to the promoter before we put that RNA polymerase back into the gene, and then it'll do what it needs to do. Same thing as DNA polymerase, it reads in 3' to 5'. So we start, there's kind of like 3 phases, and we'll talk about these phases with translation as well. But the first is the start, where we bind to the promoter. The second is the elongation, where we're going 3' to 5' and making RNA from our DNA. And then the rate of this is enhanced or silenced by transcription factors. So it's either an activator or an inhibitor. That's pretty much all you have to know. If you know those words, you kind of know the answer to that. We finish by like, do I have it here? Yeah. Sorry. So after any transcription factors have done what they need to do, the RNA transcript will kind of break off from the DNA, and the RNA polymerase will detach as well. And then we move into something called post-transcriptional modification. I can speak English. So there's kind of two big things we have to know, which are really high yield. So the methyl cap, poly-A tail, and alternative splicing. They talk about those a lot in multi-choice questions. Two other more niche things which they do talk about, but aren't as important, are the different types of RNA which affect it. So the methyl cap is added. It's like an altered, guanine, is it? Are the year twos? I can't really remember. Guanine?
Something like that.
Something
Very
like
low
that.
yield.
Yeah. Guanine. Oh, hell yeah. Cool. So guanine cap is added, and that gives a little, where does it go? Yeah. That guanine cap, which looks like the tip of this pen, is going to give a little mRNA molecule directionality. So it knows it swims forward and it can get through the nuclear pores, out of the nucleus, into the cytosol, where it can continue on to translation. The poly-A tail is just a string of not adenosines, adenines. Yeah, adenine. Who knows? I'm bad at language. Adenines, a length of A's, and that's just like if you've got a Pac-Man on the end, the A's are the pills, and the Pac-Mans are munching at those pills. And the more pills we've got on, the less likely we're going to damage that RNA. The RNA is the important information which we need to get to the ribosome so we can make proteins. Any A's on the end, that's just there to make sure that those little Pac-Man or those enzymes that are in the cytosol eating away at anything don't eat away at our RNA. Cool. The next dot point there, the product undergoes alternative splicing. So alternative splicing is, well, we get many somethings from one something is kind of the vibe of that. So can someone type that in the chat, what alternative splicing means in your own words, please? I'll have a water break then. Hydration is important as well. But yeah, so if anyone hasn't written in the chat, I'll give you maybe 10 more seconds. Aditya, awesome. Rearrangement of blocks, I'm liking that. So what we've got is, blocks is probably a good analogy. We've got a big like piece of Lego, think like Lego Masters or whatever that TV show was. That was kind of cool, better than The Bachelor anyway. So we got a big string of Lego blocks, and some of those are introns, some of those are exons. Introns, we don't really need those. Those don't contain anything that are relevant when we get outside of the nucleus. So exons, we need outside of the nucleus. They're external to the nucleus. So exons are kept, and that's really our coding region of the gene. Yeah, nice gun. Introns are interruptions. They split up all the actual coding pieces of the gene. So when we get out of the nucleus, these enzymes called spliceosomes will come along and they'll cut off our piece of free mRNA, taking out all of those introns, and so we'll get our mRNA from that. Now, the order of which some introns are cut out or which exons are included, that impacts upon the protein that's created. You can imagine if you've got like a string of AAC, AAC, A, G for one protein, you're going to get a different protein than if you've got, say, a bunch of like repeating units instead. So you're going to change the size, you're going to change the shape, and the interactions between a bunch of amino acids. So alternative splicing means we cut out introns, stitch together exons, and we can get multiple proteins from the same gene. Cool. That was a long-winded way of saying that, but we got there. So we've got two other types of mRNA which do affect post-transcriptional modification. We've got miRNA, or microsomal RNA, and siRNA, silencer RNA. So miRNA, that binds to our RNA just to inhibit it. So we've got this little piece of RNA, it'll bind to our mRNA, and it'll kind of stitch it together. It's like it's kind of trying to turn into a DNA molecule, but this holds it up until it's removed by another complex. And I've forgotten the name of that complex, but it doesn't really matter. Ricer, slicer, dicer, something ISA. Anyway, it's removed by this complex and it's held in place by miRNA. siRNA stands for silencer RNA. So if you think of like some kind of, I don't know, James Bond kind of vibe, and the villain's called the silencer because he always kills people. The silencer is the assassin. It's going to chop up this RNA into little bits and then feed it to the rice or splice or whatever, the one by one. So it chops up our RNA so it can't be used anymore, essentially. Like that nice meme I got there. Doesn't really make sense with context. So miRNA, we just bind to it. Silencer RNA chops it up. Cool. Next, translation. So when someone forgets the shape of a ribosome, classic arthememe, but that is the ribosome. So we've got our large unit on the left there and our small unit on the right. Now, initially those two units are going to be separated, but they'll come together when we initiate translation. So 3 stages. Initiation, the small ribosomal unit plus the mRNA, so a little string of genetic information, and then we'll get our first tRNA. Now, I'll hide that for now. What is the first tRNA going to be? What's the first sequence or the codon? This is fairly important. Hell yeah, Nikita. And start codon AUG. Awesome. So we'll have MET, methionine. I hate saying amino acids. The only reason I would never be able to do biomed, apart from me being lazy, is that I can't say amino acids and you have to be able to do that. It's too much effort for me. Anyway, so initiation, we have mRNA, we have our AUG codon attached to our tRNA, and that combines to our small ribosomal unit, and we get this kind of process going. Our large unit will join on top, and then we start elongating. So same as in transcription, where our RNA polymerase will kind of go along the DNA molecule, our ribosome will go along our mRNA molecule in translation. We've got 3 different sites here. So E for entry, that's where our tRNA will eventually like bind into the ribosome. That will move across to the P site, the processing site, where our amino acids bind together. Actually, did I? EPA, does it start with A, year 2s, or E? Is it entry or exit? Because it's the other way around. Epic, cool. High yield, my brain big. Anyway, so we start at A, because, okay, change the analogy in my brain slightly. We start at A because A is the start of the alphabet, P to processing, and then the tRNA will detach at E for exit. Cool, yeah. We'll change slides. Awesome. When we get to E is the termination stage. So our stop codon, which is going to be one of the 3 stop codons on the genetic code, which I don't really have to know. It might be worthwhile learning. I have to relearn them for the VIA, but it'll be right. E, so we get to exit and we get a bunch of release factors or different proteins or enzymes. They're going to bind to the final tRNA molecule and pretty much rip it from the mRNA. From there, our final amino acid is going to bind to our polypeptide chain, and we'll have this just string of amino acids. And that's the end of translation.
Before you move on, I would just like to add something. If you could go to the slide before.
Sure thing.
So in Bio 3.4, there was a question and it refers to stop codons. So a really important distinction here is the fact that stop codons do not code for any amino acids. They do not code. So if there's a question which asks you or gives you a chain, I forgot what they're called, like the AUG letters, right? So you have to find where AUG is. That's your first amino acid. And then you keep on going until you reach a stop codon. But remember not to count an amino acid for that stop codon because it does not code for an amino acid, if that makes sense.
There'll be math questions on this where it's like there's a string of 300 nitrogenous bases and it is processed through a ribosome. How many polypeptides are in there? So me being small brain, I'd think, 300 divided by 3 is 100. We'll have 100 amino acids in there. But it's 100 minus 1 because that's going to be our stop codon. We don't have an amino acid there. You'll see questions like that later on, I think.
Yeah, we've got a question from Jeff Mann. Do we need to know the mechanisms of the movement of the ribosome across the mRNA?
So when you say mechanisms, Jeff, I think the extent to which you do really need to know is just that ape. So we start at A, process at P, and then the termination occurs at E. That's pretty much it. The other thing that probably good to know is we'll have multiple ribosomes on the same mRNA. So it'll look kind of like a spiral of mRNA with a lot of dots around it as we process it. So that's just quicker, it's more efficient, and that means we can make more of the protein all at once. Does that clear that up, Jeff? I hope so.
You may also need to know when we get to energetics about the actual unit that's used for each movement. But other than that, it's not a major deal.
Cool. Yeah, so it's just, energy is needed pretty much. Let's have a look at post-translational modification. So this is after translation, we've got a polypeptide chain, and we need to do a bunch of things to it so we can make it fold and like bunch up into that protein to make it functional. So one thing, proteolysis, proteo meaning protein, lysis meaning chopping. We chop up our protein. So even though we've got some amino acids in there, they might not be necessary for the final protein. So we're going to cut those out with other enzymes just to make it smaller. Glycolysation. So glyco meaning sugar and lysation meaning adding something in. We're going to add in a sugar group to our protein. So that's for signal recognition. So if you think of like immune cells, for example, you're going to have a protein on the external surface of a cell that's going to need to like message other cells to say, oh no, there's an infection. The last one we've got is phosphorylation. So phospho, like a phosphate group, and then lation, phosphorylation, adding in a phosphate group. This is kind of how I remember it because I don't know everything going on. I'd like to use names. Nikita's raised a hand.
Are you done with the first part of your presentation now?
I've got a couple of practice questions to go.
Just before that, if we could go to the central dogma slide, like the very first slide.
Very first slide.
Yeah. Essentially, what I wanted to explain here, which I found interesting when I was in year 1, was that our lecturer told us that this used to be pretty much a central dogma. It used to be like 100%, but now they don't really think of it as 100% anymore. And the reason why is because this kind of follows DNA to RNA to polypeptide. And I think they've now discovered that there's an enzyme called reverse transcriptase, which allows you to go from RNA to a thing called cDNA. So that's complementary DNA. So just be aware of the central dogma not being set in stone. They have discovered problems with it in terms of that. Yeah.
And I think that'll be particularly important when you get to microbiome because you'll look at viruses which do use that reverse transcriptase enzyme to put their genetic material into your own DNA. I can't think of any which come to mind at the moment, but if anyone's got any, yeah, retroviruses, so HIV, any others you can think of? Anyway, that's just a general knowledge question, but yeah. Let's get a look at some of these practice questions now. So an enzyme responsible for DNA replication is DNA synthase, DNA replicase, DNA polymerase, or helicase. I'll give you a few seconds to think about that. And C, awesome, DNA polymerase. The others are kind of, well, synthase and replicase, kinda, but they're just fake. Helicase, that's our unwinding enzyme. Which of the following accurately describes an RNA in terms of sugar to the base to the strands? So this is in comparison to DNA, I suppose. Getting some A's in the chat. Awesome. That is correct. Good stuff, guys. Next, got an image, and in the section of DNA shown, which nitrogenous base is number 1? This is our base pairing rules. Okay, so I mean B, awesome, Aditya. Yeah, so we know that guanine pairs with cytosine, so we're going to have G and C, so one's going to be B. Good stuff. Next is in genes which code for a cellular protein, the introns refer to which part of the gene? The promoter region, non-coding genetic material which codes for proteins, or the signals at the end of the gene? Good job, guys. I'm seeing some B's in the chat. So non-coding genetic material is kept inside the nucleus. So in is interruptions, I think, with Khan's one, which is a good analogy. Awesome. Maybe not lastly, who knows? Role of transcription factors. Seeing some D's in the chat. So DNA transcription factors rather bind to the promoter site before RNA polymerase can bind. So we've got a bunch of different proteins. They're kind of like puzzle pieces, I guess is a good way to think about it. And they'll bind to our RNA polymerase just around it to make it either work better or worse. Cellular energetics we got here. Okie dokie.
Oh yeah, just sorry to cut in for a second.
Yeah.
Oh yeah.
I think there's only a little bit of time left in this meeting, so I'll ask everyone to pop over to the other meetings real quick.
Oh, we don't have that open quite yet, so that's fine.
Okay.
We've got some time yet since all that. Yeah.
I think it's open at
There's only 5 minutes left in this one though, so just
yeah, that's okay. Yeah, we can head in like in the last minute.
Yeah,
We'll put the link
in the chat.
Yeah.
Cool.
So just to clear something up, just before we move on to cellular energetics, so I'm not sure if you'll cover this, Will, but so before we had the question from Jeff about the movement of ribosomes, so just to clarify for those who are watching at home or will be watching later and can't see the chat, so we did clarify that the thing that you'll probably need to know is that ribosome will use GTP, which is similar to ATP, an editing molecule, and that's what's used to make it move along each space or bind a new tRNA onto the ribosome.
Cool. Thank you, Brendan. So I think, is that the last question from Central Dogma? We'll move on to cellular energetics then. Cool. So energetics. This is, I think Nirma must have lectured to you about this, and she presents a lot of information, but there's nothing that's particularly tricky. So we've got our 2 laws of thermodynamics. Energy is not created or destroyed. So anything that's input into a reaction will come out, and energy becomes unavailable upon conversion. So that's when we get a little bit of heat created between reactions. Yeah, new stream starts at 11:38. Too easy. So our total energy of a reaction is going to be H. So you might have learnt that in like chemistry in year 12. But the energy we're interested in in terms of metabolism or what the body can have is free energy, the available energy, which is given the label G. And we talk about delta G in metabolism, so the difference between the energy that's input and what is actually used. We'll talk about two reactions as well, so exergonic or endogonic, so like exothermic and endothermic. Exergonic, that's gonna release a bunch of energy, so something might be cut up, that's split in two, that's gonna release energy. Endogonic requires energy, so we're gonna absorb some energy to join two things together. The two other names for those are catabolic and anabolic as well. So we look at enzymes because enzymes are like the things which allow reactions to occur. Otherwise, we'd be waiting here for like 70,000 years for one molecule of glucose to be broken down by our bodies. So it's not an efficient process without enzymes. The three things that do affect enzyme activity in terms of environment are the pH, the temperature, and how much substrate's in there. So substrate is the molecule which the enzyme is going to be breaking down or building up. And temperature, we can see if we get to a certain point, so usually like 50 degrees, we're going to denature that protein. We're going to split the primary structure of that protein, polypeptide chain. The pH is also going to affect protein or our enzyme, and we'll have an optimum pH there. And if we fall off too far on either side, we're going to make that protein kind of fold apart because any ionic bonds or H bonds, I'm really drawing on year 12 chemistry here, sorry guys. Yeah, exactly, Nikita. But we'll get all those interactions disrupted, and so we'll get just like a blob of amino acids, but it won't actually do anything. Lastly, there's three other factors which we want to talk about as well. So enzyme activity, allosteric inhibitors are activators. So allosteric sites are just anywhere on the enzyme which isn't an active site. So molecules can bind somewhere else to an enzyme, and that will make it either work better or be worse, depending on what the enzyme does. The phosphorylation as well, so adding a phosphate group as well, that can affect activity. And the coenzymes, cofactors, or prosthetic groups are just three other things you combine onto your enzyme to make it work better. Cool, that's pretty much it. And we've got some questions, but what time does this stream end, Brendan?
Oh, okay, so we've got a minute 30
In
left
1 minute.
on this stream. Yeah.
We'll smash it out. There's
Less
only, like,
than a
20
minute,
seconds.
so yeah.
Let's go.
Yeah.
Speed round. Okay, the difference between free energy input and output in relation to metabolism is given what symbol? Sorry, I should keep that there. Getting some chats, hopefully. D, good. Delta G. So delta H is like thermochemical reactions. That's year 12. We don't really care. Which of the following factors does not affect enzyme activity? Temperature, prosthetic groups, glycolization, or phosphorylation? This is a tricky one, especially since we've got not much time left.
Getting C's from people in the chat.
Good. Glycolization, that's going to affect protein post-translational modification, but it's not going to affect our enzyme activity. Good.
All right, time to head over.
Over. Cool. See you in the next
We'll see you
internet
guys in
space,
the lab.
Bye.